सगळ्यांना स्क्रीन दिसते?
Yes, sir.
So today we'll start with oval culture technique. So this comes under organ culture. So this is again repetition for your organ. Initially 3 to 4 slides I will show you. Now organ culture is a type of tissue culture isolating an organ for in vitro growth. In this what we do, we isolate the plant organs and we grow it in vitro. So here any organ plant can be used. For example, shoot, root, leaf and flower. So in this method what we do, we preserve the structure or function. Structure means morphology of that organ and function is carrying. For example, if leaf is there, so leaf will carry photosynthesis. Ovary is there. In ovary there will be development of zygote. So here new growth differentiated structure continues given that the organ retains its physiological features. So whatever physiological features are present in that organ, it will perform those physiological features. So as such, this organ helps provide information on pattern of growth, differentiation as well as development. Now significance, it is an experimental system for nutrient and growth factor study. Then second is, we can study the interdependence of organs for growth hormones and growth factor study. Then study of morphogenesis and biosynthesis of specific metabolites. Whatever the metabolites are produced, we can study how they are produced, their biosynthetic pathway we can study, and it will open new avenues for development in agriculture and horticulture. So again tell me how organs are differentiated in two types, vegetative and reproductive. Under vegetative it comes root, leaf and shoot. And in reproductive it comes complete flower culture, isolated ovary culture and isolated ovule or embryo culture. Now what is ovary or ovule culture? Ovary or ovule culture involves development of unfertilized cells of embryo sac present in ovary. Now where haploid cells are present? Can anyone tell? Haploid कुठे असतात?
The male gamete and female gamete.
Male and female gamete. In male gamete, it is present in microspores or pollen grain, and in female, it is present in the form of ovules. Now तुम्हाला ओव्हिल आणि ओव्हरी दोघांमध्ये एकच डिफरन्स आहे. ओव्हरी इज व्हॉट inside ovules are present, and ovule is isolated unit from ovary. Means of, for example, if we look fruit of this custard apple, there are thousand not around 50 to 100 seeds of seeds are present in custard apple. So every ovule produces this seed, and the fruit itself is the ovary. But if you consider this mango, in mango only one ovary is present. If you consider jackfruit or furnace, there are many ovules because many seeds are found in this jackfruit. For example, jamun is only one seed is present. So here what happens in basic difference in ovary culture is we culture whole part containing ovule. So अख्खा आपण ते जे ओव्हरी आहे ते कल्चर करतो आणि ओव्हिल कल्चर मध्ये काय करतो, वी ब्रेक दिस ओव्हरी and we isolate the ovules and we culture these ovules. So एवढं डिफरन्स लिहायचं. when there is a short note, what happens if you they will tell you describe ovary culture, मग तुम्ही अख्खा ऑर्गन ड्रॉ करायचा आणि we have isolated the ovary. आणि काय करायचं, we have break this ovary and isolated the ovule and took this ovule as an explant. So here is the typical structure of flower. So can you see this ovary and ovule? सगळ्यांना दिसतंय का?
Yes, sir.
शाळेचा जरा आवाज दिसलं का सगळ्यांना?
हो सर.
Then ovary. Hello.
Hello, sir. This is
Now you can see how well protected this ovule is there. So this is protected first with calyx, then corolla, then again stigma. Inside the stigma, this ovary is there, and so in natural fermentation, fertilization, what happens? Pollen grain is the egg cell in the ovule to produce zygote. बरोबर ना?
yes, sir.
So this, as this seed is very important, it is highly protected and aseptic conditions are maintained. So while selecting, what happens for ovule culture, what we say, we select the unopened buds are selected, flower buds, because inside this microbial contamination is very less. We will see this in protocol. Now you know the position of ovule and ovary. Now what is meaning of ovule culture? In this ovule culture is an elegant experimental system by which ovules are aseptically isolated from ovary under control conditions. So what happens in ovule culture? The ovary is aseptically dissected, and from this ovules are removed. And these separated ovules, they are grown on chemically defined nutrient media. So it will contain MS salts and growth hormones under controlled conditions. So this is definition of ovule culture. Similarly, what will be the definition of ovary culture? In ovary culture, the ovary is isolated aseptically, and whole ovary containing the ovules are grown on a chemical defined media. Now the principle behind this is ovule is a megaspore integument. Ovules are attached with placenta inside the ovary by means of funicles, and ovule contains a megaspore or egg cell. This egg cell, after fertilization, forms cell zygote, and which ultimately leads to formation of mature embryo possessing shoot and root primordia. So if you consider this groundnut, when you open these two cotyledons of the groundnut, inside you can see small leaf-like structure. That is nothing but embryo. So this is the formation of fertilization between male and female gamete. So the haploid part before fertilization, ovules are haploid in nature, but after fertilization, this zygote is formed. Then ovules can be isolated and cultured in nutrient media. In vitro, ovule culture helps to understand the factors that regulate the development of zygote. Now, if you want to study the zygote, what you have to do? You have to dissect this ovary and study the after fertilization how zygote is formed. So you cannot look at all stages. But in culture, what you can do? You can isolate this ovule and you can induce it to produce zygote. And how zygote form you can study. Alternatively, it may be possible to germinate pollen in the same structure as exile ovule and to induce in vitro fertilization and subsequent embryo production. Then what is the protocol here? First, what you have to do, collect the open flower or unfertilized oils. If the oils are desired, collect the open flower where the anthers are the highest and pollination has taken place. How you will know the pollination has taken place or no? The flower is open, stigma tries to dry. Here, what happens? The pollination has been completed. But if you want unpollinated, then what you have to take? You have to take flower bud. To ensure the fertilization, collect the flower at 48 hours of anther dehiscence. Anther जे आहे ते ड्राय होतं 45 hours after anther dehiscence नंतर हे आयसोलेट करायचं. Then remove the pulse, petals, androgen, etc. from the ovaries containing the fertilized or unfertilized ovules. Then for this disinfection, what you have to do, soak the ovaries in 6% NaOCl solution. This is sodium hypochlorite solution. Now it is a bactericide, so it will kill any contaminant bacteria present. Then rinse ovaries 3 to 4 times with sterile distilled water. This is the sterilization process. Next, using sterile techniques, ovules are prodded with the help of spoon shaped spatula by breaking the funicles at its junction with placental tissue. Now these ovules, they are connected to placenta with funicle. So you have to break this funicle. So you'll get separate ovaries. Sorry, ovules. Then the spatula with the ovules is gently lowered into the sterile solid or liquid medium as the culture vial is slanted about 45 degrees. So what happens? We will provide this semi-solid medium and we will put this slants into 45-degree angle. So we'll get slanted media, solidified media. And in this slanted solidified media, you will dispatch these ovules. Damaged or undesired ovules are rejected when possible during transfer. What happens? During breaking, what you'll do? You some ovules they will get damaged. They will be crushed. Cell sap will come out. If you are keeping it outside for a longer period, it will get browning will take place due to phenolic secretion. So the such type of ovules are rejected. Now this is our inoculation process. Then what you have to do? Incubate the ovule culture in either dark or light. 16 hours, 3000 lux, light intensity, and temperature is 25 degrees Celsius. So this is the protocol. See, here is the plant. From that plant, you have to the flower. So which flower you have to take? The anther should dehisce, means dry, and after that 48 hours. This flower you select, you will wash with distilled water, dip in 5% tipol, then again wash with distilled water. Then you will do sterilization with 5% or 6% sodium hypochlorite solution. And this is a laminar airflow chamber. In this laminar airflow chamber, what they have did, they have separated the ovary. And they have put this ovary directly on the media. And from that, you have got, after 2 weeks, haploid plant. आलं लक्षात? प्रोटोकॉल आला का लक्षात सगळ्यांच्या?
हो सर हो आलं.
So see, this is the protocol given for ovary culture. Ovule culture मध्ये काय करायचं फक्त? You have to break the ovary and separate the ovules. सगळ्या स्टेप सेम आहेत. So this is the example. See, तुमच्या A figure मध्ये before dissection the flower. B मध्ये काय केलेलं आहे? ओव्हिल्स आयसोलेट केलेले आहेत आणि मध्ये जे डी फिगर आहे की नाही त्याच्यामध्ये बघा ते ओव्हिल जर्मिनेट व्हायला लागलंय. आफ्टर जर्मिनेट इट इज स्टार्टेड डिवायडिंग. मग डिव्हिजन झाल्यानंतर काय झालं, दे हॅव गॉट ए प्लांट ई. एफ मध्ये काय केलं त्यांनी, ते सेपरेट केलं, अजून ग्रो केलं प्लांट एफ मध्ये. आणि जी मध्ये काय केलं, यू गॉट ए होल प्लांट. आणि एच फिगर मध्ये काय केलं, दे हॅव आयसोलेटेड द सेल्स अँड स्टडी द नेचर ऑफ हॅप्लॉइड और डिप्लॉइड. सो यू कॅन सी द क्रोमोझोम इज हॅप्लॉइड हिअर. डिप्लॉइड असेल तर 2 सेट असते क्रोमोझोमचे. ही फिगर लक्षात आहे का सगळ्यांच्या? सगळ्यांना फिगर कळाली का?
हो सर.
हॅप्लॉइड नेचर कळालं सगळ्यांना. इन एच फिगर दे हॅव स्टेन्ड दिस क्रोमोझोम. ते क्रोमोझोम सिंगल सेट फक्त दिसतोय, हेन्स इट इज हॅप्लॉइड. This is a microscopic method to study the haploid or diploid nature. So now what you have did, you have studied the principle, definition, principle, protocol, and I have given you example of this flowering plant. Now we'll study the applications. Now why this ovary or ovule culture are important? So isolated ovule culture as early as zygote or 2 or 4 cell pro-embryo stage is of considerable importance. Why? Ovule culture is in obtaining seedlings from crosses which are normally unsuccessful because of abortive embryos. See, every time the zygote formed, it will not produce seeds. It will abort. Abort म्हणजे काय करणार, disintegrate. मग abortion थांबवण्यासाठी फ्लॉवर मध्ये what we can do, after fertilization 4 cell pro-embryo stage we will isolate that and we'll culture it on a semi-solid medium. Hence what happen, abortion का होतं, the plant finds that it is not useful or the plant is not capable of providing the nutrients. Hence this embryo will get killed. But here what we have got option. Before this embryo has been killed, we can isolate it and we can take it in culture. म्हणजे जे प्लांट डेव्हलपच होणार नाही, वी हॅव डेव्हलप्ड इट. सो प्लांट ब्रीडिंग मध्ये काय होतं, efficiency is very less, but we can increase this efficiency and we can get novel crosses by using this ovule culture method. First application is test tube pollination and fertilization. The technique of test tube pollination and fertilization. By this technique, it may be possible to germinate in the same culture as egg-side ovule and to induce in vitro fertilization. We take this ovule and we put pollen also together and we induce in vitro fertilization. म्हणजे जे आर्टिफिशियली होणार आहे ते आपण काय करतो, इन विवो ते इन विट्रो करतो. So we can study in which the morphological changes or biochemical changes and structure how the cells divide in using this test tube pollination method. जसं आपल्याकडे ह्यूमन्स मध्ये टेस्ट ट्यूब बेबी, इन द सेम मॅटर दिस वर्क्स फॉर प्लांट्स ऑल्सो. दिस एक्साइज्ड अनफर्टिलाइज्ड ओव्हिल्स ऑफ एक्झाम्पल कुठल्या प्लांट्स मध्ये दे हॅव ट्राइड, आर्जिमॉन आहे, पेपॉवर आहे, सोटॅनम आहे. ह्या स्पीसीज मध्ये काय केलंय, दे हॅव कल्चर्ड दिस पोलन ग्रेन्स विथ रिस्पेक्टिव्ह ओव्हरीज and all the stages of development starting from germination of pollen to double fertilization have been observed, and the mature seeds containing viable embryos have been obtained by the above experiment. आता डबल फर्टिलायझेशन होतं हे कसं कळेल आपल्याला? You have to study in vitro. How they have the pollen tubes enter, how it will fertilize with the double fertilization, egg आणि synergid cells. तर ह्या सगळ्या शकतो. So due to this technique we can do. Using the same method, it has been possible to fertilize ovules of Melanandrium with pollen grains from other species of carrier of IC. आता प्रॉब्लेम काय? पहिल्यांदा मी तुम्हाला सांगितल्याप्रमाणे अबॉर्टिव्ह एम्ब्रियोज तयार होतात. तर ते एम्ब्रियोज थांबवण्यासाठी वी कॅन यूज दिस आणि क्रॉस पॉलिनेशन पण एक दोन जवळचे स्पीसीज आहेत त्याच्यामध्ये आपण क्रॉस पॉलिनेशन करतो. फॉर एक्झाम्पल दिस potato plant or tomato plant that come under same family Solanaceae. So you can fuse these potato pollen grains with tomato embryo. Normally असं होईल का? पोटॅटो पोलन ग्रेन आणि टोमॅटोच्या एगला फ्यूज करेल? नाही होणार, सपोर्टच करणार नाही. बट इन आर्टिफिशियल मेथड वी कॅन डू दिस. Employing ovule culture technique, the incompatibility barrier in Petunia axillaria has been overcome. Abortive embryos किंवा इनकॉम्पॅटिबिलिटी आहे. काही काही प्लांट्स मध्ये काय होतं की egg after fertilization will not survive only, it will produce only vegetative. तो पेट्युनिया सारखा, तर त्याच्यामध्ये ओव्हरकम केलेला आहे या मेथडने. Now application of ovule culture in hybridization. So in many interspecific and intergenic crosses, the F1 hybrid embryos frequently become abortive in developing seeds are not capable to support the development of embryos. ह्या केसमध्ये काय करतो? आर्टिफिशियल ओव्हल कल्चर घेतो आपण, ओव्हल्स आयसोलेट करतो आणि आफ्टर फर्टिलायझेशन अँड वी प्रोव्हाइड इट विथ न्यूट्रिएंट्स, इसेन्शियल न्यूट्रिएंट्स. कुठले असतील? एमएस मीडियामध्ये शुगर्स असतील, 16 मायक्रो एलिमेंट्स असतील, मॅक्रो एलिमेंट्स असतील, विटॅमिन्स असतील आणि हार्मोन्स असतील. आणि वी कॅन गेट दिस अँड फ्रॉम दिस एम्ब्रियोज वी कॅन डेव्हलप द होल प्लांट. म्हणजे हायब्रिडेशन होत नाही. हिअर वी कॅन प्रोड्यूस दिस हायब्रिड. तर हे कुठं कुठं ट्राय केलंय? ए एस्क्युलेंटा इंटू ए फिनिकस या स्पीसीज मध्ये, किंवा ए एस्क्युलेंटा इंटू ए मॉस्किरेटस आणि ए ट्युबरक्युलेटस इंटू ए मॉस्किटस ह्या वेगळ्या स्पीसीज मध्ये इंटरस्पेसिफिकली त्यांनी हायब्रिडेशन केलेलं आहे. सिमिलरली a true hybrid between Brassica chinensis and Brassica pekinensis has been obtained by culturing fertilized ovule in vitro. Now a hybrid between Lolium perini and Festura rubra has also been obtained successfully by means of ovule culture. Now several attempts have been made to hybridize between different species of the new and old world cotton. तर कॉटनच्या ज्या जुन्या स्पीसीज आहेत आणि ओल्ड मधल्या स्पीसीज आहेत जुन्या आणि न्यू, त्यांच्यामध्ये पण हायब्रिडेशन केलेलं आहे, पण ते नॅचरली होत नाही. पण ह्या ह्याच्यामध्ये झालेलं आहे. त्यामुळे तुम्हाला काय होतंय? कॉटन वाय यू आर प्रोड्युसिंग कॉटन फॉर इट्स फायबर. मग तुम्हाला फायबर क्वालिटी चांगली वाटते, प्रोडक्टिव्हिटी मिळते जास्त. So what happens? In vitro they have produced fibers and they have studied the various roles of hormones or phytohormones on fiber and seed development. हे पण त्यांना कळालेलं आहे. Then next come production of haploid callus to ovule culture. Now if you want haploid cells, you have to go for pollen grains or ovule. These pollen grains you get in maximum quantity as compared to ovule. So Uchimiya et al. In 1971 attempted culturing unfertilized ovule of Solanium melogene and obtained various callus formation on media supplemented with IA and kinetin. Now, for example, you want to go for haploid callus. What you have to do? Select how you have to select the ovule. Unopened bud घ्यायला लागेल. बिकॉज ओपन बड असेल आणि त्यामध्ये जर अँथर जर ड्राय झाला असेल तर त्या केसमध्ये काय होणार? fertilization झालं. But in unopened bud, what happened? Fertilization is not done yet. Hence, what happened? The ovule cells are unfertilized. Unfertilized means they are haploid in nature. On haploid plant side, you have to take explant as a bud, flower bud, unopened bud. Although the origin of the callus tissue was not known, a cytology assay revealed it to be haploid in nature. मग तुम्हाला कसं करणार? माझे मी फोटो दाखवलेला सायटोलॉजिकल क्रोमोझोमचा फोटो. त्यामध्ये काय करतात? दे टेक दिस कॅलस सेल्स and do staining for nucleic acid material and they observe under microscope. So reveal the nature it is haploid or diploid. So by this we can obtain haploid cell line or plant from alternate sources rather than anther or pollen. Instead of using anther or pollen, you can use from ovule culture. The next is ovule culture and angiospermic parasites. It is generally believed that the obligate root parasites such as striga or orovanki, the formation of seedling is dependent on some stimulus from the host roots. So studies on ovule culture of orovanki and tristanki have demonstrated that the formation of shoots in vitro can be induced in absence of any stimulus from the host. So these parasitic plants, if you see this mango बघितला कसं मँगोचं झाड, त्यावरती पॅरासाइटिक प्लांट असतात बघा. आपण त्याला मराठीमध्ये पांडुळ म्हणतो. So parasites they will not grow on ground, they will require host only. मग आतापर्यंत समज काय होता की फॉर डेव्हलपमेंट ऑफ ओव्हिल they need host root system or shoot system. पण ते काय केलं आपण इन विट्रो कल्चर करून ते चुकीचं सांगितलं की देर इज नो नीड ऑफ दिस host system, root or shoot system for the development. Then ovule culture of orchid plants. In nature, the seeds of orchid germinate only in association with a proper fungus. Orchids, जर तुम्ही हे बघितलं तर orchid it is epiphytic. It will grow on a substrate. It may be bark, huge trees, stone or wall. हे orchid होता. आणि त्यांचं association कोणाबरोबर आहे fungus. Name of that fungus is mycorrhizas. So this mycorrhizas it will produce water, nutrients यांना देणार. आणि orchid will produce food and shelter. As a result, numerous seeds are lost due to proper fungus. प्रत्येकच फंगस अप्लाय मिळेल असं नाहीच असोसिएशन होईल. म्हणून काय होतात जे सीड्स तयार होतात ते लॉस्ट केले जातात. सो बिसाइड दिस द सीड कॅप्सूल ऑफ मेनी ऑर्किड टेक्स ए लॉंग टाइम टू मॅच्युअर. सीड फॉर्मेशन झाल्यानंतर 6 महिने ते सीड कॅप्सूल डेव्हलपच होत असते. मग इन दॅट पिरियड इफ एनी पेस्ट इट विल हॅव और बर्डनी खाऊन टाकला तर त्यांचं प्रोपोगेशन होणारच नाही. To overcome such problems, attempts have been made to culture this fertilized ovule of orchid in vitro. म्हणजे ओपन फ्लावर ओपन झाल्यानंतर अँथर ड्राय झाल्यानंतर ते काय करतात, ते रिमूव्ह दिस ovule and they have cultured it and they have did this in Calanthea vetium, Cypripedium insigne, Dendrobium species and Palaemnosis scilariana. हे जे ऑर्किडच्या स्पीसीज आहे त्यामध्ये हे सक्सेसफुली प्लॅन केलेलं आहे आणि ह्या मेथडनी ऑर्किड प्लांट्स डेव्हलप केले जातात. Then next example application is induction of polyembryo by ovule culture. आता हॉर्टिकल्चर प्लांट मध्ये एक पॉलिएम्ब्रियो होल्ड्स अ व्हेरी इम्पॉर्टंट ग्रेट पोटेन्शियल. पॉलिएम्ब्रियो आपल्याला मँगो मध्ये मिळते. सी whenever you consume these mangoes and you throw away these mangoes, what happens? It will produce the seeds. Seeds produce करेल आणि these seeds will germinate. But what happens? Some seeds they will produce only one shoot and some will produce 10 to 15 shoots. This is called as polyembryo. So nucleus of this monoembryic ovule like citrus can be induced to form advantageous embryo in culture. Therefore such achievements are very significant. म्हणजे काय वेळेला आपल्याला जिथं एकच आपल्याला एम्ब्रियो मिळेल तिथं आपल्याला पॉलिएम्ब्रियो मिळेल, त्यामुळे मेनी शूट्स मिळतील. तो हे खूप महत्वाचं आहे. नाव आपण बघितलं तर why we go for tissue culture? One of the application is disease free material. म्हणजे बनानास आपण काय करतो, टिश्यू कल्चर करतो त्यावेळेस आपण काय करतो, एकदम त्या बर्डच्या आतमधलं इनिशियल मटेरियल घेतो, बनानाच्या रूट रायझोम मधला. त्यामध्ये काय असेल तिथं व्हायरस नाही, बॅक्टेरिया नाही, पॅरासिट नाही, काय नाही, त्यापासून आपण स्टार्टिंग मटेरियल घेतो. सो हिअर there is optimum condition for there is no presence of virus and bacteria. But citrus species मध्ये काय येतं, लिंबू, लेमन, ऑरेंज, संत्री, मोसंबी, देन पपनस वगैरे हे जे फ्रुट्स आहेत हे सिट्रस फॅमिलीमध्ये येतात. इट इज अ व्हेरी लार्ज फॅमिली. तर ह्याच्यामध्ये काय होतंय की व्हायरस इज अटॅकिंग एव्हरी पार्ट. तुमचं जर शूट सिट्रस लिंबू, इफ यू हॅव सीन दिस लेमन प्लांट, त्यामध्ये काय होतं, जास्त प्रोटेक्शन नसतं न्यू शूट्सला जेव्हा नवीन लीव्ह डेव्हलपमेंट होतं, त्या तिथे काय असतं, त्यामुळे व्हायरस एंट्री इझिली होती. मग तुम्हाला जर व्हायरस फ्री प्लांट मटेरियल पाहिजे असेल, व्हॉट यू विल डू, आफ्टर फर्टिलायझेशन ऑफ दिस फ्लावर you will isolate the ovals and you culture the ovals to obtain virus free material. So this is one of the example. Last one is the anther and pollen culture. So by careful selection of developing anthers and critical stage, it is possible to establish anther culture that will give rise to haploid plant. Alternatively, the pollen grain can be diverted from their normal pathway. Somatic काय करणार, पोलन ग्रेन्सच काय करणार, आपण सोमॅटिक एम्ब्रियोज तयार करायला लावणार आणि ती सोमॅटिक एम्ब्रियोज विल प्रोड्यूस germinate into shoots and roots and we can get plants. So haploid plants will have single set of chromosome. The phenotype is the expression of single copy of genetic information and they will have only single characteristics. Haploid plant मध्ये तुम्हाला मागे मी सांगितलं, समजा if you take P for tallness, tall they will give capital T and dwarf will get small t. So if you double T, capital T, plant is tall, and if the plant genome has capital T and small t, then also the plant is tall only, heterozygous. But here all these genes are homozygous because it will contain only single set. So breeding program मध्ये आपल्या plants हे फार महत्त्वाचे राहतात. So in this matter of completed our ovals and ovary culture. फक्त तुम्हाला एवढं लक्षात ठेवायचं आहे की ओव्हल कल्चर मध्ये तुम्हाला जर नोट आली ओव्हल कल्चरची, तर तुम्हाला काय करायचं आहे, यू हॅव टू डिसेक्ट द ओव्हरी अँड isolate the ovals and you are going to culture the oval, and in oval culture what happens, whole ovary is cultured. एवढं फक्त प्रोटोकॉल मध्ये डिफरन्स लिहायचं आहे. आलं सगळ्यांच्या लक्षात? हॅलो,
Yes, sir. Yes, sir.
आलं ना? कुणाला काही डाऊट्स आहेत का?
नाही सर, नाही डाऊट.
बाकीचे कोणाला काय? थांबायचं मग आपण. हॅलो,
Yes, sir. Yes, sir. चालेल.
मी पीपीटी शेअर करतो तुम्हाला.
हा सर, चालतंय.